Can you hear me okay? That's pretty loud. I try to start my sessions right at the hour because it works well with the recording system. I'm just noticing though, see those little red circles up there on the ceiling? Those are supposed to turn green when the recording system is on. So if those don't turn green, can you hear me okay without this? Is that okay? Okay, it's gonna. Could I? Okay, all right, that's fine. Anyway, if those don't turn green, I'm gonna step outside and talk to the IT. I think there probably is, but I'm more likely to make things worse by pushing things if I push the wrong thing. So I'm a little reluctant to do that. Okay, it's 11:01 by my watch. Would you just give me one second, please? Thank you. That's it. Okay, so it was recording.
They won.
You are recording.
It was.
Fire hose begin.
Okay.
Down here, that's what it's going to record.
Okay, super. Thank you.
Yep.
Okay, everything's all sorted out there. Hi, welcome. How's everybody doing today? Good. It was great to see you all at the white coat ceremony on Friday. Hope you all enjoyed the weekend. Did you? Okay, good. So yeah, that's the most stressful part of medical school is out of the way and under your belt. So now we're going to have, now the fun begins. So my name is Andy Skildum, and I teach medical biochemistry here. I have an office and lab up on the 3rd floor, so if you ever want to meet me, that's where you'll find me if I'm not in the classroom, and the various ways to contact me. You know about session objectives and the significance thereof. Okay, so this is what you should be looking at before you start studying a session, because this is what you'll be tested on. Okay, and everybody's got the point solutions on their phone. Okay. All right, well, let's start. So first of all, I want to say welcome. Thank you for being here. I'm very happy that you're all here. We need awesome doctors in Minnesota, and you're well on your way to being one of them just by setting foot in the door here. So thank you. Glad you're here. Okay, so the purpose of this little brief session today is just to get a little orientation for where we're going in the medical biochemistry content over the next almost 2 months of the Foundations of Medicine course. So the basic outline, we're going to talk about some protein structure and what an enzyme is and how an enzyme makes biological reactions proceed under a regulated and very precise fashion. So once we get that out of the way, we'll talk about central metabolism, essentially how we gain energy from the food that we eat, how we store excess chemical energy in our body to make it available for when we're not eating, and etc. Then we'll talk about the details of carbohydrate metabolism, amino acid metabolism, and fat metabolism. And we'll be focusing on the oxidation pathways and the storage pathways and some of the biosynthetic pathways. But a lot of the biosynthetic pathways will be covered in further courses. Okay. In terms of the resources for the course, has anybody got the Mark's medical biochemistry textbook yet? Not yet? Okay, I would encourage you to get a paper copy of the Mark's Basic Medical Biochemistry. It's a good book. I've used it for a long time, and it's a resource that you'll use for the 2 years of your undergraduate medical education curriculum. I think you'd also find it useful for board preparation. So I'm using the 5th edition for all of my figures and whatnot. So I'll have figure numbers and page numbers on all my lectures, and those will refer to the 5th edition. There's a new 6th edition that just came out this summer. I haven't had a chance to look at it yet, so that's available if you want to use a 4th or a 3rd edition. The content is largely the same. You'll just have to do a little extra work to get the page numbers and figure numbers to match up with the way that I present it. So yeah, it's a good book. If you want the most expensive one, you get the 6th edition. If you want to save some money, you can get a 4th or a 3rd edition, and it's the differences will be minimal. Okay, fair enough. If you don't want to get it, I think you can get like some older editions online even. So a lot of options there. Okay, we'll also be looking at using the Stanford pathway of human metabolism, which is, I don't know if you can see my dot over there. And this, I'm not going to be teaching directly from this, but where the different content appears on the map, I'll illustrate it, and it's a good reference for you to kind of have a comprehensive overview of the different pathways that we'll talk about, and it also is nice because it shows how the different pathways are interrelated. So that copy is going to be posted in the room here for indefinitely. If you want, you can get a printout to put up in your own study area, and I don't know, they'll charge you $20 or $30 in the IT department if you want one. So that's available for you. Let's see, the University of Minnesota Health Sciences Library, that is a wealth of resources are available through that link. So this is a great place to start any type of a search that you're doing for review or independent research or what have you. That's a great place to start. Has anybody used PubMed before to do a literature search? Okay, so if you do PubMed searches through that link, then any article or any journal that the university has a subscription to, it's like it'll automatically know that you have a subscription and make it available. Okay. Let's see. So for the general format, you know about Canvas and how the sessions are listed in Canvas, right? Okay, so I don't want to go through that in detail. Objectives and the reading assignments, those are posted well ahead of time, and I think for all of my sessions for Foundations of Medicine, that's all available for the whole course now. So if you want to go ahead, you certainly can. In terms of the PowerPoint downloads, don't look for those generally until, say, later in the afternoon of the day before a lecture. That's kind of, sometimes I might be earlier than that, but it's kind of the way that I work. I like to wait till the last minute, I guess. Okay, so we'll also have some review questions or practice questions, and these will be kind of interspersed throughout the lecture. I don't give you these in the download ahead of time, but right after class, I'll post those for you to have them available for your review. Okay, and as far as attendance, it looks like, boy, just about everybody's here today, a full house. Probably not going to be like this on Wednesday is my guess. But so attendance for these lectures is not mandatory. Everything is going to be recorded, and in the rare occasions that the recording systems don't work, then we can pull a last year's recording or something. So I think it's fun to be in class live. That's why I do this rather than record them. But if that's not your thing, you definitely don't have to be here. So, okay. Let's see. This is the detail of the schedule. Oh, this is the textbook, Amazon link, the Stanford pathway. I guess I got a little ahead of my slides here. So, University of Minnesota Health Sciences Library. Okay, any questions? Is everybody signed into your session with that Turning Technologies app? Should we test that out a little bit? Okay. See how that works. See how well you know Duluth.
Oh my gosh, you're probably not cheating.
You're from Duluth, right?
I'm not cheating. Academic integrity. Think about what to do.
Okay, does that everybody figure out how to enter in an answer? And I think you can change your answer up until the last moment if you want to.
It was on the first. No, I was right.
Hey, very good. Is that 100%?
Almost.
Wow, great job. Yep.
Can you go back to the session ID? Is that
Yes.
Yeah.
Yeah.
Can you read that? 588276. You're welcome. Okay, so after class, as soon as I get up to the office and have a few moments, I'm going to put like this slide and all the other quiz slides in a separate PowerPoint for download. And that's going to be the format for all of my lectures. Okay? All right, so we got near 100% on that one. What do you think about this one? Okay, good. So they're going to start out a little easy and get harder as they go, or get more relevant at least, maybe not harder. Okay, this is pretty straightforward one. The peptide bond, which is a type of an amide bond, right? Okay, getting a little harder. Okay, getting really probing the depths of amino acid chemistry knowledge. And just take your best guess for these. It's okay, super. So aspartate and glutamate, that ATE ending is kind of consistent with being an acid that's ionizable, right? This is generally 100% question, but see on this one. No pressure. No, yeah, no pressure. And this is just, giving you some example of like what the lectures are going to be like. Not that you're expected to know all this content. Okay, secondary structure, very good. Okay, this is an example of a more difficult question and something that by the end of this course you should have no trouble with whatsoever. Do you want to talk with your neighbor about this one for a moment?
And not
And I had to bring my macros today. I looked at it. I just.
I mean, it could be.
Okay, so Reddit runs like my blood cells and you give those nutrients.
The idea.
Okay, you want to select your final best answer and see if we have a class consensus? Everybody got a chance? Looks like we got 54 people have responded, 55. Ooh, okay. So I think the best answer would be folic acid. So a lot of people picked that. A few people picked cobalamin, vitamin B12. Did you pick that one? No, I didn't. Who picked? Well, that's the second best answer, but there's something in this question that would steer you away from that. Okay, so this is a tough question, I think, if you haven't had, nutritional biochemistry in your background. So I'm going to go through a little bit more kind of content about some general principles, and then in the conclusion of the class, we're going to pick this question apart and use it as an example of how, if you don't just know it right off the bat from intuition, about how you can approach these more complicated questions in a systematic way. Okay, fair enough? Okay. All right, so let's talk about a few key concepts. We'll talk about the three principal macronutrients, talk about anabolism versus catabolism, and then fuel oxidation or oxidation states of carbon. And then I think after those three are covered, then we'll go back to some biochemical problem solving. Sound good? Okay. So what I have next are just some slides that are, in my several years of doing this, lecturing and foundations, just some things that get everybody onto the same page and give us a little bit of unifying vocabulary that we can use to talk about some of the things that you'll hear repeated over and over and over again throughout the class. And the first thing that we'll talk about are the macronutrients. So macronutrients, the definition of a macronutrient is, I guess, the way I see it, it's something that you can use to get ATP from, right? You can burn it to get energy from. That's different from a micronutrient like a vitamin. A vitamin, is essential for catalyzing the reactions that oxidize the macronutrients, but you don't get a lot of caloric value out of a molecule of vitamin C, for example. Okay, so that's different from the macronutrients. So the macronutrients are the carbohydrates, fats, and the proteins. Okay, or monosaccharides, disaccharides, and polysaccharides for carbohydrates. Fat is typically in the form of triacylglycerol. That's our main dietary fat. And then amino acids are the kind of building blocks of proteins. Okay, so I don't want to get ahead of myself here, but let's talk about carbohydrates first. So carbohydrates refer to sugars, so these kind of intermediate oxidized molecules that have equal numbers of oxygens and carbons. So if we have just a single subunit, this is referred to as a monosaccharide. And the monosaccharide glucose, this is like the the dollar bill of our body. This is the most common form of currency that every cell can accept, and every cell can use it to, buy half a cup of coffee or make some ATP from. Okay, so all of the other carbohydrates, whatever form they're in order to be used by the cell, a few exceptions to this, but they're converted into the monosaccharide glucose. So the glucose is the most important monosaccharide, the most important carbohydrate that circulates in our blood, but we really don't eat glucose as part of our diet. Glucose just isn't really there unless you're taking an oral glucose tolerance test or something where you have a drink that's engineered to be all glucose. So most of our carbohydrate energy comes in the form of polysaccharides like starch, shown here. So amylopectin or amylose are the two principal starches where we get most of our carbohydrates from. We also get some carbohydrate energy from sucrose, the disaccharide, the table sugar, and then from lactose, the milk sugar, is also a disaccharide. Irrespective of where they come from, they're all converted into glucose in our body, and then glucose is the carbohydrate that is taken up by the majority of our cells and can be used for energy. Okay, and I'll just mention so glucose is what's oxidized for energy. If we're eating carbohydrates in excess of the energy needs of the moment, and we want to store excess carbohydrate energy, we'll store it in a molecule that's very similar to the starch. This molecule is called glycogen, and it is a branched polymer of glucose that's very similar to dietary starch. We can also convert excess glucose very easily into fat, into a fatty acid, and store it as triacylglycerol. So as you're looking at these macronutrients, kind of think about where they are in the diet, what their fuel oxidation pathway is, and what the storage pathway is. So those are kind of things to keep in mind. Okay, so further on to the macronutrients, let's talk about protein. Protein is an important source of calories in our diet. However, protein has the problem that it contains nitrogen. So there's one nitrogen atom or more for every amino acid, and there's no caloric value for nitrogen. We can't oxidize nitrogen to get fuel. And in fact, we have to expend a little bit of energy to convert the nitrogen into urea in order to get rid of it, to keep it below toxic levels in our body. So for that reason, the protein has a lower caloric value if you consider how much ATP you could get from each mole worth of fuel. Protein also is, I would say, it's the most diverse because we have 20 or 21, depending on how you're counting the amino acids, different chemistries at this R position, and you have then multiple different pathways that you can use to oxidize the amino acid side chains for using as a fuel or converting to some other biosynthetic process. Okay, so let's see. The form of protein in our diet is generally as a protein or a peptide where individual amino acids are bound together to make a protein. And then during digestion, those peptide bonds are cleaved, and then individual amino acids are brought into the blood. If we're doing a ton of work right at the moment that we're eating that peptide or eating that protein, these amino acids will be oxidized down to carbon dioxide and also to urea and used to make ATP to meet the energy needs of the moment. If we have excess protein in our diet relative to the immediate needs of our body, we will then synthesize protein and use the proteins both as essential little machines that do things that are useful in our body. But proteins are also the storage form for amino acids, for excess amino acids in our body. Okay. Okay, and then the final macronutrient that we'll consider are fatty acids. Fatty acids, these are the most calorically dense fuels that we have in our diet. And they're also the most calorically dense fuel stores within our body. Okay, so fat, you get quite a bit more ATP per mole or per carbon with fatty acids being oxidized as you would for glucose or for protein. And that has to do with oxidation states of carbon, and I think I'll introduce that in a few slides here. Okay, so the main form of fat in our diet is as a triacylglycerol. So the triacylglycerol, the basic structure is you have a 3-carbon alcohol of glycerol. That's kind of the scaffold. And then at each one of those 3 carbons of glycerol, you have a fatty acid on it. Okay, and that fatty acid can be either a saturated fatty acid shown here or an unsaturated fatty acid with one or more double bonds between the carbons. So triacylglycerol, this is where most of our fat calories come from. However, we also can have significant amounts of fat calories from membrane phospholipids, for example, where you would have 2 fatty acids and then a phosphate linkage and then an acetal or serine or whatever for a membrane phospholipid. Yeah, so those are the main dietary forms of fat. To get energy from those fat, from that dietary fat, we have lipase enzymes in our gut that will cleave off individual fatty acids from the glycerol backbone. These fatty acids then are rejoined into new triacylglycerol and then distributed throughout the body, either oxidized for the immediate needs of the work that we're doing right at the moment, or they can be stored as triacylglycerol in adipose tissue. Okay. I'm not sure if I mentioned cholesterol here, but cholesterol is an important molecule that comes along with fat, and it's very important for the physical properties of fat-containing membranes. It's a very different looking molecule, and it doesn't get oxidized for fuel like fatty acids do, but the metabolism of the two are intimately linked. So maybe just put a star and think about cholesterol along with this. And we'll have a lecture a month and a half from now on cholesterol. Okay, everybody got the 3 macronutrients down? Okay. So let's discuss anabolism and catabolism. Okay, so if we think about metabolism is everything that we do. And if we wanted to divide that or categorically divide that, we could divide it into anabolic reactions or catabolic reactions. So catabolic reactions are, as I mentioned before, if we have a molecule of glucose and we convert that into 6 molecules of carbon dioxide, that's one large molecule going into much smaller molecules. That's a catabolic reaction. Okay, so fuel oxidation reactions are catabolic processes. Generally, they're making small stuff out of large stuff. Okay. Now, if we have excess glucose around and we wanted to synthesize a large polymer like glycogen, that then would be an anabolic reaction, a building reaction. Okay, you're making a larger thing out of smaller subunits. So Dr. Cormier probably talked about DNA synthesis just Well, was he asynchronous or was he live? Okay, so if you haven't seen it yet, DNA synthesis would be an example of an anabolic reaction. Okay, so just kind of keep those two concepts in mind. They're somewhat linked to fed state and fasted state metabolism, which we'll talk about in next week, I think, in a little bit more detail. But essentially, when we want to get energy, when we're doing a lot of work, we're going to be doing catabolic reactions because those catabolic reactions are ATP generating generally and can power mechanical work or biosynthetic work or what have you. Then when we have excess fuel around relative to the work that's being done in the moment, then we're going to generally be doing anabolic reactions or storage reactions as shown up there. Now, this is, I guess, the case for an adult who is, neither gaining mass or losing mass. If you're thinking about like a developing child, it's a little bit different. Okay, so catabolism versus anabolism. Let's talk, I think, well, I've got a few other key concepts here. We'll talk about fuel oxidation and oxidation states of carbon as the next key concept. Did anybody take physical chemistry? A couple of you? Okay. It's of no use to you whatsoever in medical school. So yeah, you could probably expound on this topic for a long time, but this is all you're going to see about valences from me, and I'm definitely forgot way more than I know now. Okay, so fuel oxidation. I'm going to go back a little bit. Oops. Okay. If you look at this fatty acid, for example, or just maybe it's easier to see here. So this fatty acid is made of multiple carbon-carbon bonds, right? And those carbon-carbon bonds or what defines a carbon-carbon bond is that those carbons are sharing electrons in their outer orbitals, right? And that sharing of electrons makes a covalent bond between those two carbons. Now, the carbon can be either reduced or oxidized depending on how many of those electrons are in that outer shell. Okay, so if you have a single bond, as shown here, where two carbons are sharing two electrons, but the other three are not shared, you can pull two electrons away, and then the two carbons that are bonded are sharing four carbons, and then there's only two unpaired electrons around each carbon. That would be a double bond. Okay, so that pulling electrons away from carbon, that's key for fuel oxidation reactions. And the thought here is that the amount of potential energy in the carbon at this state is higher than it is at this state. That allows the reaction to generate energy that can be used to power subsequent reactions. Now, I have shown here these electrons just kind of going off into space, and that's not really how it works. Every oxidation reduction reaction has a redox partner, and those electrons will be bonded to those redox partners, and I'll expound on that process in a moment. Okay, so essentially we eat carbon in the very reduced form, such as a fatty acid that would be shown up here. Through the processes of fuel oxidation in our body, so fatty acid beta oxidation or glycolysis and what have you, we're oxidizing the carbon further and further and in a orderly stepwise process to generate carbon dioxide. And the carbon here is at a lower potential energy than it is up here. So we can use the energy that's lost at each one of these steps to power other reactions or do mechanical work or what have you. Okay, so you'll hear me talk a lot about alcohols and aldehydes and carboxylic acids. So those are different oxidation states of carbon, and the methyl or the alkane that has more potential energy or more electrons than does a carboxylic acid. Okay. Have you all seen that before? Some of you? Okay, good. Okay, so when we eat fuel, we are taking our diverse sources of macronutrients and we're stripping electrons away from carbon-carbon bonds and we're making smaller little bits. Now, most of the fuels that we will consume in our diet have as a common intermediate the acetate short chain fatty acid. So acetate is a 2-carbon carboxylic acid, and the acetate, the 2 carbons here, were present in our dietary fuel. Okay, so a 6-carbon glucose molecule that can be broken down into 2 carbon dioxides and 2 acetates ultimately. Right? So the acetates don't just exist freely in the cell. Well, there's a little bit, but in order to be used as a fuel oxidation intermediate or a biosynthetic intermediate, it's bonded to a molecular handle called coenzyme A. So coenzyme A is not the fuel part of this reaction. It's the handle. It's the way the enzymes grab onto the fuel. Okay. All right, so with glycolysis, for example, the 6-carbon glucose gets broken down into 2 molecules of acetyl-CoA. The 2 carbons from the acetate here, these can get converted into carbon dioxide through the tricarboxylic acid cycle, and then the fuel is completely oxidized. Right. Now, that's where the carbon went. Where do these electrons go? Right. So we're losing electrons up here. We're also losing electrons from the carbon in the TCA cycle. So those electrons are transported through oxidation reduction cofactors like the nicotinamide adenine dinucleotide or the flavin adenine dinucleotide that act as electron carriers that will carry the electrons to the electron transport chain, and that then is the site of ATP generation in the mitochondria. Okay, clear? Okay, so I want to just talk about that molecule of acetyl-CoA again because it's so important. So acetyl-CoA, now acetyl-CoA is an intermediate in the oxidation of carbohydrates, in the oxidation of fats, and in the oxidation of most, but not all of the amino acids. Okay, so acetyl-CoA is almost a universal intermediate in the fuel oxidation, but some amino acids can be oxidized independently of acetyl-CoA. So what acetyl-CoA looks like, the structure is shown here. So remember the 2-carbon acetate, this is the part that is a component of our diet, like is a component of a fat or a carbohydrate or what have you, that gets converted, broken down into the 2-carbon little unit here. And these are the carbons that are going to be converted into carbon dioxide and oxidized completely down there. So the rest of what you see here is just the way that different enzymes grab onto that acetate. Okay. So this structure is the adenine nucleotide, so the adenine base, ribose sugar. Then we have this portion here. This is derived from pantothenic acid or vitamin B5. And then you've got the acetate portion. Okay, so just to demonstrate here, acetyl-CoA is a common intermediate in the oxidation of fuels and diverse fuels from our diet. The acetyl-CoA then, this can, in the context of a cell that is doing a lot of work and has a high requirement for ATP, the acetyl-CoA or the acetate portion of it will be oxidized into carbon dioxide in the tricarboxylic acid cycle. If we're in a situation where we're not doing a lot of work, we can then take some of that acetyl-CoA and use that as a feedstock for different biosynthetic reactions within our body. Okay, now shown here, so we can do a lot of this in our body, but we can't make vitamin K in our body. So like this, some of the reactions shown here are plant reactions and bacteria reactions. Okay. I'm going to go back a slide or two. Okay, so remember back, and this is a figure that, you know, if you have a good printer or a printer, this is a good one to print out and tack up on the wall in your study area, okay, because it's just easy, and basically we're going to be breaking apart each of these little steps over the next coming weeks and expanding on it a lot. So this is a nice map that shows where we're going. Okay. So the next key concept that I want to introduce is the oxidation reduction cofactors. So these electrons, as these are stripped away from carbon, they don't just exist as a little E minus out in space. Okay, they're way too volatile for that. So these electrons have to go onto a different organic molecule and to reduce it, and then that different organic molecule can get oxidized to release the electrons to a different substrate down the line. Okay, so those electron transfer cofactors, I think I'm going to go forward one and then I'll come back to that. Okay, so this is similar to that other cartoon. This one adds a little bit more detail showing the role of the redox cofactors. So we have some oxidation reduction cofactors that are generated in the cytoplasm by through glycolysis, for example, in the glyceraldehyde 3-phosphate dehydrogenase reaction. So the nicotinamide adenine dinucleotide, NAD, this is, I guess, well, you're going to hear me say this in just about every lecture. So this is a very important oxidation reduction cofactor. NADH, this is the reduced form. So the reduced form has extra electrons on it. Okay, and the electrons come from the carbon-carbon bonds in our dietary fuel up here. Okay, so the NADH then, the reduced form of it, this is a way that electrons can be carried to the electron transport chain in the mitochondria. The NADH then can be oxidized by complex 1 of the electron transport chain. Those electrons then are carried along the electron transport chain, and ultimately they're used to reduce molecular oxygen down to water. Okay, that's like the least amount of potential energy, so that's where the electrons are going. And then the energy difference at each one of those steps is captured and used to power ATP synthesis. Okay, so just want to show you the structure of the NADH, the nicotinamide adenine dinucleotide. Shown here, let's see. So I don't test on structures necessarily, but there are a few that you should kind of be familiar with, and I think this adenine nucleotide is definitely one of them. Like, so this is the adenine base, the adenosine base here, and then the ribose sugar is in the plane here, so that's harder to see, but anyway. So this part of the molecule is easy for the body to get. It's just adenosine nucleotide. This part of the molecule, though, this is derived from a vitamin. So this is something that we can't synthesize ourselves. We need to eat the nicotinamide or vitamin in order to synthesize this. So this is one example, and I'm not sure if I've maybe make a note of this, of a link between macronutrients and micronutrients. Okay, in order to get the caloric value of a macronutrient like glucose, we need to have micronutrients so that we can synthesize these redox cofactors. Because if we didn't have those, then we can't take the electrons from the substrate that we put in our mouth and bring it down to the electron transport chain. Okay. Okay. Is that clear? Clear as mud? Okay, are there any questions?
Could you just rephrase that last bit one more time? So you need micronutrients to synthesize cofactors to get energy from macros?
Yeah, so like when we're making this, when we're reducing the NAD plus to NADH in the fuel oxidation, we're then reoxidizing it back to NAD plus down here. So this NADH, this isn't, we can make 1 million molecules of glucose through this pathway down to carbon dioxide. The NADH doesn't get consumed, it gets kind of recycled, okay? Flipping back and forth between the oxidized and the reduced form. So we need a very tiny amount of the nicotinamide, just enough to make that little electron carrier to flip back and forth between the oxidized and the reduced form. And that tiny amount of NADH, that's going to facilitate the whole flow of the cycle and allow you to burn a lot of your macronutrients. Is that clear? Other questions? Okay. All right, why don't we return. I'm going to have to go through this stuff pretty quick, but hopefully it'll be useful. Okay, remember this question? What was the right answer? Folic acid? Okay. All right, so how do we solve biochemistry problems? I'm going to just, like I said, I'm going to try to end on time, so I'm going to go through this quick. Okay, first thing, read the last sentence first. Probably heard this as advice before, for especially if you have a long paragraph here. Before you read this one, and that kind of tells you what to look for in the big stuff. Okay, then scan the answer choices. That might give you a theme for what to look for. Okay, now all of these things, well, except for metformin, that's kind of an oddball, but everything besides metformin that could reasonably be associated with an anemia. Okay, 3 out of the 4, the folic acid, niacin, and cobalamin, those are nutrient-derived things, right? So maybe this is about nutrition. Okay, so then let's break it down. So presenting concern, this is like what brought the patient in to the clinic or the emergency room or the specialist or what have you. Now in this case, it's fatigue, and that's very nonspecific, like anything can make you tired, but it suggests a problem with energy metabolism, right? It's also interesting that it's progressive, right? It's not just stable, but it seems to be getting worse. Okay. Let's look at the history. So this is less likely to be a glycogen storage disease or Lesch-Nyhan disease or something like that, because this is a middle-aged adult who is coming in with this concern, right? A lot of inborn errors of metabolism tend to be diagnosed in, young children, right? So just the basic demographic parameters of the patient, that can kind of lead you towards one thing or another. Then you would look at diet history or kind of lifestyle issues. Smoker, abuser of alcohol, smoking maybe, increased cancer risk, more oxidative stress. Alcohol, that can be kind of a lot of things with alcohol. Increased oxidative stress. fatty liver, the whole bit. Okay, so let's look at the laboratory analysis next, and we have a macrocytic anemia. Okay. It doesn't mean that there weren't a lot of other laboratory tests done, but this is the one that's, significant. Right. Okay. Would you just stay with me for 2 more minutes? Okay, I'm gonna try to go through this quick. You have all these slides in your download, right? Okay. Okay, so look for a normal physiological process that is disrupted. Okay, don't focus on the pathology so much, but try to figure out which normal thing has gone wrong. Okay, is it a problem with nitrogen handling in the urea cycle, for example? Is it a problem with nucleotide recycling, something like that, right? So there are normal processes that have substrates and then make products. And when those things are functioning normally, you don't have symptoms. And when they're disrupted, then you do have symptoms. And the specific symptoms that you have are related to the normal function of the pathway. Okay, so in this case, the normal physiological process would be red blood cell homeostasis. So anemia, that means there are too few red blood cells. That's the definition of anemia. Okay, now how many red blood cells we have in our body, this is going to be a balance between erythropoiesis, or the generation of red blood cells. It's going to be reflective of their experience in the circulation, and then it's going to also be reflective of the rate of destruction of the red blood cells in the spleen. Okay, so you could have an anemia because of delayed erythropoiesis, or because of bleeding, or because of overactive red blood cell destruction. Now in our case, we have a macrocytic anemia. So we have too few red blood cells, but the ones that are there are too big, right? So I'm just trying to think of whether I should try to rush through this or if I should just complete this on Wednesday. I think I'm going to complete it on Wednesday because it's a good habit to be in time and keep to the schedule. So I'm going to try to live by that. Okay, so yeah, it's too much to rush, and I don't want to rush it. So take a look at it on your own in between now and Wednesday, and I'll take 10 minutes out of Wednesday's lecture, and we'll go through this, and hopefully it'll be useful for us. Okay. All right, I apologize for being off schedule already on your first lecture. Okay, if you have any questions on the material so far, come and see me now or see me in the office upstairs or send me an email. All right, thank you.
Oh, really? I'm like, I wonder if he remembers me, because I remember. I did not feel related. Go, go, go. Get out of here. Yeah, it was hard to
Yeah, and like, clicked back, it was like, he was like very nice and kindly, had a good conversation, and I was like, I'm gonna, yeah, you can read up, yeah. That's what I would do too. Every day I was like, do you remember me?
I'm gonna go home after this and eat some food and that's way
That's like,
I think that is.
I mean, it's like, at this point, like I finished the first 2 lectures.
I was like,
I don't know. Yeah.
But I think that's the main thing that I struggle with, just yes, okay, I'm sending you. This is the form objective outline for each.
Yes,
Oh, she did, okay, perfect.
yep, I'm already here.
So I think the best thing I'm gonna do